Um,
Yeah, you can go ahead. Sorry.
I can go ahead.
Yeah.
Good morning. My name is Tanya, and I'm Franco's mentor. So I've been working with Franco this last weeks that he's been with us at Steinhauser Lab. Not only me, but Franco has been interacting with all the lab members, and it was a pleasure to have Franco with us. So since the very first day when we were talking about the projects that we have at the lab, and we are a quite diverse lab, Franco was asking questions, and he was actually asking really smart questions. So it was very promising from the beginning. And then Franco just engaged in one of the projects that we have in the lab, which is studying, we are trying to investigate new regulators of bone loss. And one of the means that we do that is by fasting. So we study the effects of fasting on bone. And Franco just jumped into this project. It has been over a year project. So Franco just helped us with that by conducting an experiment. And this is the experiment that Franco will be presenting today. So I hope that Franco had fun with us. We had fun. He's always welcome to come back. So Franco, good luck, and the floor is yours.
Okay. So once again, my name is Franco Alvarez. I was in the Steinhauser lab this summer in Bridgeside Point 1 in the Aging Institute. My mentor is Dr. Tanya Amorim, and my PI was Dr. Matthew Steinhauser. And this summer, my project was studying the effects of methionine on osteoblast differentiation. So like my mentor already kind of glossed over a little bit, all this started whenever we were researching fasting and the effects of fasting on the body. There's a lot of research showing promising benefits in terms of fasting to many areas of the body, and a couple chronic conditions such as obesity, diabetes, cancer, and hypertension. But one of the things that we were more focused on was the negative effects of fasting on bone health and how it can seem to cause a lot of bone loss. We want to look at how we can maybe reverse that or stimulate bone growth. After a 10-day fasting study, we discovered that P1NP, which is a bone formation marker, decreased in healthy patients. And inversely, we saw that the essential acid methionine actually increased, which was kind of our first clue to how we could possibly treat this bone loss phenomenon. So that was our goal this summer, was to just dive into that and study it, see what's going on. And we wanted to find out whether methionine would change how bone forms. We were studying positive or negative impacts for that matter. And just because I think everyone here is on a little bit different of a level, I'm just going to start off with the basics on bone. There are three major types of bone cells: osteoblasts, osteoclasts, and osteocytes. Osteoblasts are important to us because they are the bone forming cells. That is what we really wanted to study because we're interested in healing bone and increasing bone mass. Osteoclasts, although they sound intimidating, are still very important to bone health, because if you don't have a balance of osteoblasts and osteoclasts working together, that's when you start having imbalances in bone. You can have bone loss, too much bone, and so you definitely need all the cells working together to keep bone healthy. And osteoblasts, like I mentioned, are the cells responsible for forming bone. This is the differentiation process. This summer, we studied just the effects of methionine on this process right here, from pre-osteoblasts to mature osteoblasts. This was about a 16-day process, and by the end, we had mature osteoblasts if the treatments worked well. Like I mentioned already, when osteoblasts malfunction or they don't work properly, you get that imbalance in bone, and that can lead to bone loss, which is why we are so interested in studying why osteoblasts stop working, or how we can treat them so that they start working better. So overall, this summer, my aim in my study was to investigate the effects of methionine on osteoblast differentiation. And my hypothesis from the P1NP and methionine levels that we saw at the beginning was that excess methionine decreases osteoblast differentiation. So this summer, this was the exact setup I had. I had 2 6-well plates set up exactly like this. 4 different treatments: no methionine, 20 micromoles, 100 micromoles, and 500 micromoles. We plated the exact same amount of human pre-osteoblast cells. It's from this cell line that probably doesn't mean a whole lot, but if you're a science person, it does. We plated the same exact amount of cells in each well, And then we added 4 different treatments of methionine, and this was included in the media. So as we changed the media every 3 days, the cells had fresh food to kind of grow from. They differentiated in a 39.5 degrees Celsius incubator for 16 days, like I mentioned, until they were fully differentiated. And at the very end, we pulled the plates out, stained it with alizarin red, which stands for calcium deposits or mature osteoblasts, because it's all kind of tied together. And we just looked at which wells had the most alizarin red for the most amount of differentiated osteoblasts. On the final day, we took some microscope pictures. These are what our wells looked like. I forgot to include this for the people that were here last week with me, but this is pretty important to look at because you can actually see how no methionine really affected the differentiation process. And there's a lot less cells compared to the other 3 treatments. It's hard to see with the eye between the other 3 treatments, and even with the next picture too, with the staining. But the most important thing you can take away from the pictures, I think, is that no methionine heavily impacted the way that osteoblasts differentiate. And there was a lot less like cell proliferation, and there's just not many cells left by the end. So once we did the readings, we found that the most significant data was that pre-osteoblasts treated with no methionine were significantly less like proliferating by the end of the differentiation process. And excess doses of methionine did have less differentiation, but whether this data was completely significant, we would need to do more research on to figure out whether our hypothesis was completely correct or not. You can also see that 20 micromoles was the optimal level for our osteoblast differentiation. But the most significant thing to look at from this bar chart, I think, is that no methionine was the largest inhibitor in whether osteoblasts were able to differentiate successfully. So when it comes to the end, methionine clearly does have an impact on osteoblast differentiation. We saw that in our study, we needed an optimal level of about 20 microliters. And it does appear that osteoblasts do not differentiate well in the absence of methionine, which is different than our hypothesis, which we may need to do more research on to figure out whether that's true or not. Like I said, our hypothesis was never really like disproved or proved, but our lab is continuing to do more research on whether this effect is true in not only cells, but also biological systems like mice and other systems to see if what we see in the cells is true in the actual body. And yeah, that's all. We have time for questions.
Maybe you remembering wrong, but I think you call methionine an essential amino acid in one of your.
Yeah.
What does that mean?
Well, essential amino acids are used to create proteins throughout our body. So whenever methionine is used to create proteins, those proteins could be used to help our bone, I think. I know methionine is like tied to the start codon for most proteins. So methionine is really important in making proteins, but in terms of our study alone, we were just looking at how methionine affects osteoblast differentiation. Yeah. Well, we're doing a current study on mice to see whether these effects are true within the actual like living, breathing organism. We're still working on that. Yeah, that would be really important in looking at what the optimal levels are for osteoblast differentiation. I only had 4 treatments just to keep it kind of simple. Now that we kind of saw our results and saw that 20 microliters was like the most optimal level, we might do more research to see like find the balance of exactly which levels are the most optimal. But that would require more research, obviously. Did you have a question? I'm sorry, I didn't hear you.
So I noticed that the treatment was carried out at 39.5. What is the reason for doing
That starts the 39.5 degrees Celsius is the temperature that premature osteoblasts begin to differentiate. I want to pull up the slide just to point it out again. To start this process right here at the beginning, that's where the 39.5 degrees Celsius comes in, because that's what starts this whole process. That's where the cells are most happy to continue growing, these specific types of cells. The only thing I studied this summer was actual cell work. So as far as applications in the whole body, I'm not too familiar with.
Good job. All right, Sophia, you're up.
Yeah, there's some
Yeah, the projectors will get you. Here.
Okay, so my name is Dorota Jaswinska. I work in the Cervantinakis lab, and I had the pleasure of working with Sophia this summer. She got a little bit of an introduction to bioengineering, since that is what she wants to do whenever.
Apparently, this is the one before his.
School.
Okay, is that all right?
Yes, I'd like to see it.
Is somebody talking to us? And mute. Mute. Maybe that one. I don't know why we can't mute that person. Mute.
I guess. Sorry for that interruption. So Sophia got a little bit of an introduction to bioengineering and medicine because she wants to go to medical school later on. And we looked at ovarian cancer metastasis, since that is my thesis project, and she took a little bit of it and looked at how MMT works with ovarian cancer. So thank you.
I think I'll stay. So as Dorota already introduced, my name is Sophia Alvarez, and I was in the Zerva lab this summer, which is in the Center for Biotechnology and Bioengineering building, and it's literally the one like 2 doors down. And again, this summer I studied the effects of mesothelial to mesenchymal transition on high-grade serous ovarian cancer metastasis. I'm gonna just press the next one. Okay. Sorry. So just a real quick overview, ovarian cancer. Every year annually, there are 21,000 women in the US diagnosed with it and about 14,000 deaths on average. Ovarian cancer is the 8th most common type of cancer, but the 5th leading deadliest cancer. Often it's not diagnosed until advanced stages, which is what makes it so fatal because we can't catch it at early stages, so it's hard to treat and stop. The most common and deadliest type of ovarian cancer is high-grade serous ovarian carcinoma, which is the cancer that we were studying this summer. And high-grade serous is a type of epithelial cancer that originates in the fallopian tubes, which these are the fallopian tubes here. One of the ways that high-grade serous is able to metastasize is through the peritoneum, which just lines the abdomen, as you can see over here. This peritoneum is made up of a monolayer of mesothelial cells, which is one of the key points that we studied this summer. And as you can see here, if the primary tumor is here, it can break off and go through the mesothelial layer that makes up the peritoneum and metastasize to other parts, as you see here throughout the body. Another focus point that we had was MMT, which is just mesothelial to mesenchymal transition. And mesothelial cells, as I already mentioned, they often form monolayers in the body around vital organs. And mesenchymal cells are more stem-like and more motile, so they want to move around more. And if you've ever heard of like mesenchymal stem cells, they're usually the cells that differentiate into other types of cells in the body. And you can see here MMT shown, where here are the mesothelial cells, and you can see them transition into mesenchymal cells over here. One of the main ways that we were able to study this summer was through microfluidics, which is like the bioengineering part of it a little bit. And this is the top view of the devices that we had. They're very little, probably only about like this big. And some advantages of using a system like this is that it's shown in 3D rather than 2D, and it allows for cells to be placed in a certain way. So rather than in a plate, just kind of putting cells on top of each other, we can actually like place them in places such as these channels here to watch how they interact with each other, again, rather than just being kind of mixed together in a plate. So as you see here, the channels, this is where we put the mesothelial cells, and we can put cancer cells in there as well. And then this gel region has collagen in it, and you can watch the cells actually kind of move around and invade and just kind of see how they interact. And these posts here are just to kind of keep that gel in so it doesn't leak into the channels. And then again, why we used it is because it simulates an in vivo assay, and also can predict similar outcomes to an animal without actually having to use animals. And this is from a different paper that worked with breast cancer, just to show you the 3D component of it. You can see that here would be the channels where we put the cells, and this is the middle channel wherever we'd have the collagen in the cells, and whatever else is in there can move around, just to kind of give you a visual of that. So our hypothesis was that high-grade serous cancer tumor cells can transform mesothelial cells to mesenchymal cells, and that this interaction between the tumor cells and mesothelial cells allows for increased metastasis. Here's one of the first things we did, was just tissue staining. The lab actually had like in vivo experiments in the mice, and so they cut up the tissue and put it on slides, which I was able to stain and actually look at. So the first thing that we do is deparafinize the tissue, which is essentially kind of removing the wax, and you start it with xylene and move it to lower grades of ethanol, so that way the stain will actually go into the tissue. The next part is quenching, which Jordan explained to me is kind of like boiling the tissue, and it literally looks like a pressure cooker, and we have to write not for food on it because it looks like a pressure cooker. And then after that, we block the tissue. This is to block non-specific targets, so that way whenever we stain, we're not staining all the cells in the tissue, we're only staining for the cells that we want to look at. And then we add the primary antibodies, which attach to the cells you're looking for. So if we're looking for mesothelial cells, then the primaries would attach to those cells. And then you have the secondaries, which attach to the primaries, and the secondaries are the ones that have the immunofluorescence on it, so you can actually see the color whenever you image them. And then lastly, we just image the slides. So here's one of the pictures from the tissue staining, just to kind of show you what it actually looks like. This in yellow is mesothelin, which is a marker for mesothelial cells. And you can see, as I mentioned, it does form a monolayer and kind of just like encapsulates the cells here. And then the blue is DAPI, which stands for nuclei and cells. So it just kind of shows you where the cells are. And now again, the main thing that we did this summer was work with microfluidics. So to make these, we had to make PDMS molds, and it's just a 3D mold, and we pour PDMS in, and then we punch holes. The holes would be these ports right here, so that way we can actually put the cells in, and again, everything else we're trying to put in. Then we have to autoclave them and bind them to glass, so that way we can actually look at them under a microscope on a glass slide. Then we coat them with poly-D-lysine, which this just allows the collagen to actually adhere to the glass better in the middle, so that we don't have cells in between the glass and the collagen looking like invasion, but it's not actually invasion. And then we fill the center channel again with collagen type 2, and then coat the channels with fibronectin, and the fibronectin just allows the cells to stick to the channels. Then we add the cells, and then for some of the assays, we treated them with TGF-beta, which is just an MMT like signaler. So we would add the TGF-beta 24 hours after the cells if we were going to add it in. Here is one of the assays where we put mesothelial cells in the top channel, which is the green here. And then we put tumor cells in the bottom channel here. So we have mesothelial in the top two, and then tumor cells in the bottom. And this line is OVCAR 9 or OVCAR 8, sorry. And then this top picture is from June 21st. This is when we first seeded the cancer cells. And you can see that there is a little bit of invasion here that kind of went through the post a little bit, but 8 days later on June 29th, you can actually see the invasion towards the tumor cells is greater. This is another assay we did where we actually put the cancer cells in the same channel as the mesothelial cells to see if they would invade through, kind of simulating again that peritoneum and the monolayer where it goes through. So over here, you can see the mesothelial cells with the cancer cells in red here, and you can see that they're going through. And just to show you that they're not just on top of the mesothelial cells, but they actually invaded through, you can see over here on the right, there are the holes where the tumor cells actually like invaded through. Again, this is another similar picture to the one I just showed you. This is in a different device that we did. Again, here are the tumor cells and the mesothelial cells here. And then you can see those holes from where they actually like invaded through and made those holes. And then this one was a picture 72 hours after we seeded the cancer cells. This is using the TGF-beta that I kind of mentioned. So cancer cells actually give off TGF-beta signaling just kind of naturally in the microenvironment, and we think that it induces MMT, which would allow the cancer cells to be able to metastasize better. So here's our control without TGF-beta, and you can see that the mesothelial cells are like more circular, the monolayer is kind of more flush and they're stuck together. But over here are the mesenchymal cells that have been treated with 10 nanograms per milliliter of TGF-beta, and you can see there's like holes here and they're more like clumped and they're not as like smooth. And that's because again, they're more motile and they want to move around and they don't want to like stick together and just be flush. So our conclusion is that the OVCAR-8 cancer cells invaded through the mesothelial monolayer, that the TGF-beta treatment induced morphological changes. Also, I forgot to mention with the MMT, you could see the structure was different with the cells. Here, I'll go back really quick. The structure was different with these, but to definitively be able to say it induced MMT, we'd have to stain for mesothelial and mesenchymal markers, which would be like one of our next steps. But so far, we can't see morphological changes. So we saw that. And then preliminary data suggests that the mesothelial cells invade towards the cancer cells. And then future directions, just adding cancer-associated mesothelial cells, which have gone through MMT. So taking those cells that have already gone through MMT and then mixing them with cancer cells to compare them to the normal mesothelial cells and see if it makes a difference. And then also staining for mesothelial and mesenchymal markers for MMT, which I mentioned again, just to definitively say, yes, MMT happened. And then also possibly knocking down TGF-beta signaling to slow metastasis. So if it makes it easier to go through the mesenchymal cells, actually stopping the transitioning from happening would make it a little bit harder for the cancer cells to go through. And then just a huge thank you to everyone that supported me this summer, like the entire lab, Dorota especially for spending all the time with me this summer, Dr. Duncan, Serafina, and then everyone at the Hillman site, and some of my references. So any questions?
On Zoom,
Oh, yeah.
question, will you just repeat the question?
Yeah.
And also the Zoom people are welcome to ask a question too.
Yeah.
So your full strength.
We're basically just seeing how they're getting through because the actual logistics of how they get through isn't really studied. So we don't know what to like strengthen about it. We're also just looking at MMT because it does occur. So we were like seeing if there is a difference and if it makes the cancer cells better. So right now we're kind of just studying like the invasion and like seeing how it actually goes through because nobody really knows. Yeah. You mean like, oh yeah, she asked like what substrate I use to mimic the peritoneum. Do you mean like the fluids that we put in? Oh yeah, that was collagen type 2, which is actually really abundant in the body naturally. So that's what we use. The part that mimics the peritoneum is actually that mesothelial monolayer. But again, since the collagen is already abundant in the body, we use that so that way it kind of just is like a middle part if the cells want to move through it or not. Does that make sense? Yeah. Mm-hmm. Yeah. Like almost like the flow of it if they're actually able to get through. So we did have trouble with some, but honestly with the devices we had, it was pretty consistent. So if the flow didn't work, we did have devices where they stuck in the port or they only went halfway through. They're essentially just kind of trash and we didn't use them for actual data. Okay.
Do you
Yeah.
think, as a mechanism, it's recruiting the cancer cells? You think it's TGF beta, maybe the dominant mechanism?
That's like making them be able to metastasize better.
Right.
Yeah, we think that's what we're testing out because it already kind of occurs in the body. So we think that that might actually be one of the reasons why it's able to metastasize. So that's why we're studying it.
Do you think that's the only mechanism? Or are there other
No, I think there's other signaling. I forget what they're called, but we studied other one and there were markers that we studied for other signaling, but I don't think TGF beta is the only one. Yeah, we just wanted to see how big of an effect it had. Thank you. This up here.
Yeah, you guys are welcome to, get coffee and all that in between the talks. That'll be free. Who's gonna do the introduction?
I'll do the introduction, but I could probably just.
Yeah.
However.
Whatever. You call it up. Wait for everybody to sit down in a second. Okay.
You know what I say for like
If you just stand in front so they can see you.
We don't get the shot here, so. Hi, good morning, everybody. My name's Travis Lear, and along with Anya Boudreau and Dr. Bill Chen, we've had Elise Chu in our lab for this summer doing research. And Elise came in with a little bit of wet lab experience, but she had a really strong determination, great work ethic, and by the end, she was doing protein assays and cell work like crazy. So it was great. She's done a great job. And over time, we have been really thankful to have her in the lab. She's come in, she's gotten a crash course in molecular biology and taken that with a lot of patience, determination, and a great attitude. That's really brightened the whole lab. And we've been really thankful to have her here. And we really can't wait to see what you do next. Wherever you're going to go, it's going to be great. So it's been a real pleasure to have her here, and we'll take it away. Give it to you here.
Where'd it go? Okay. I'm sorry about that. Hi, everyone. My name's Elise Chu, and this summer I was joining my lovely mentors and my PI at the Chen Lab in Ridgeside Point 1 at the Aging Institute. So we were researching how the ubiquitin system can be used to hinder COVID-19 stability. So my PI was Dr. Bill Chen and my mentors, Travis and Anya. So we're focusing on limiting COVID-19 replication. So just for some background, the coronavirus replicates by hijacking a cell in the lungs because it's a respiratory infection. And then it makes one of its ribosomes make the genomic protein of the virus. So in this case, it makes a chain of RNA right here. And then it cuts up that chain using a protease. So each little bit that's cut off turns into a new virus. So we want to focus on this protease, and the one we're looking at specifically is called NSP5. The other large component we're looking at is the ubiquitin system, which is used to degrade proteins. So for example, if a protein has completed its function or if it's damaged, then the ubiquitin system kills it out. So first, ubiquitin ligase attaches to the target protein, and then an enzyme is used to activate the ubiquitin protein, which then attaches to the target protein to make a polyubiquitin chain. And then that chain binds to a proteasome that then degrades the target proteins back into amino acids. So in our research, we used OC43, which is a less dangerous type of coronavirus that contains a protease that's similar to the one in COVID-19 called NSP5. And then to host these viruses, we're using Bs2b human bronchial cells. And in Bs2bs is the human ubiquitin ligase E3. And then our techniques that we used is HiBit, which is basically a luminescent tag that's used to measure how much protein is in a sample. we use a Western blot, which basically separates proteins by size. So we can see how much of each protein is by how thick each individual band is. And then we use this thing called transfection, which is to artificially infect cells with specific plasmids. And then we use this stuff called CHX called cyclohexamide. So it's basically a cell culture tool that's used to stop cellular translation. So the way I was taught, if the cell was a tub and the water that's coming in are different types of proteins, including the virus, CHX basically stops the faucet and stops any protein from producing. So our first objective was to basically prove that ubiquitin had an effect on NSP5. So we used a luminescent tag, the high bit tag that I talked about before, to attach to NSP5. And then we treated wild type and the E3 knockout Bs2Bs, so basically cells with and without the E3, to with the CHX for different times. So you can see like for 0 hours and 3 hours. And then we looked at how much NSP5 degraded with and without the E3. So on this blot, this is a immunoblot that I did at the beginning of the summer. So basically we have the cells with the E3, so we can see there's a huge decrease in the NSP5 that we're looking at versus the sample with the cells without the E3. There's a much less or a much smaller decrease. So from that, we can kind of conclude that the E3 was responsible for a lot of the NSP5 degradation. And then our next objective is finding the ubiquitination site on NSP5, so where the E3 attaches to NSP5. So first of all, ubiquitin attaches to a protein by making a peptide bond with an amino acid. So if this is NSP5 and these are, I don't know if you can see them, they're like little colorful bands that are branches that are poking out. One of those branches that are poking out is going to be where the ubiquitin ligase attaches. So we've been mutating different amino acids on NSP5 and then doing CHX time courses like in the other experiment, but only with the ones with E3. And then we do this thing called pull down to basically isolate how much ubiquitin there is. So if the NSP5 levels are constant, that means that there's little to no ubiquitination. So that would indicate that the site that we mutated is a critical ubiquitination site. So these were some of my preliminary results from doing a transfection. So this is a CHX time course. I don't know if it shows up very well on the board, but you can see little timestamps on the plate for when I transfected stuff. And so these are the different NSP5 mutants that we did. And then we basically put it in this opaque plate and then put it in a machine to read luminescence, which you can see over here. So this is essentially how much NSP5 there is in each well. And then we looked at the average rate of change from 0 hours to 6 hours, and we found that this K to R mutation had the least amount of change or the least amount of ubiquitination. So we looked more into it, and these are different experiments that we did. This was like kind of confirming that the mutation had less ubiquitination. So this is with the wild type NSP5 and the mutation NSP5 in different CHX time courses. So with the normal one, we can see that the level of NSP5 decreases significantly versus the one with the mutation stays pretty much constant. And then we did this other thing called a CFC time course, which is basically, if we go back to the cell being a tub, CFC is the drain, so we see proteins accumulate. So with the wild type ones, we can see that this that would represent ubiquitin increased a lot versus the one with the mutation didn't really have any decrease, which means that there wasn't any ubiquitin protein or little to none ubiquitin protein. And then to finally solidify our findings, we did a pull down to find how much ubiquitin there is. So with the wild type NSP5, we can see that there's this huge blot of clogged protein right here, which is the ubiquitin. So regularly we would see this, but with the mutation, we don't see any types of clumps like that, which indicates there's little to no ubiquitin. So this is just a lot of results saying that the mutation that we have has like less ubiquitination. And then I just wanted to shout out to the Chen lab. Everyone's really cool. My mentors are awesome. I'm gonna miss the lab tech Elise, my other half. But yeah, and then that's it. Any questions?
Am I allowed to ask a question?
Yeah.
Ah, super. Elise, this is awesome. Your experiment is so much work. I am so impressed. Something I was curious about when you were talking is this this NSP5, is this like something that's common to other viruses as well, or is this something unique to COVID?
So for coronavirus, they all have that protease, which is called 3CL protease. But the one specifically for OC43 and for COVID-19 is NSP5. Did that answer your question?
Yeah, I was wondering if your technique could extend to other viruses, if there are other similar NSP5s in other viruses, or is this really focused?
I think it can't, like the techniques that we were using can definitely be applied to different hypotheses. I'm pretty sure it would be like different sites, there would be different nuances, but the experiments and techniques can definitely be applied to different viruses.
Wow, that's really great. You did an amazing experiment.
Is this Dr. Waldeck?
Yes,
Oh
it's
my
me.
Hi.
But I have to leave now because Michael Ulyss is speaking in another Zoom call.
Oh yeah, at the surgery. Okay, have fun.
Nice job.
Yeah. Any other questions? Yeah.
I like the ubiquitination, the way the ubiquitination is really cool, where you found the sites and you're looking at the degradation of the protein over time. And so was it the K48 that you think is the important one?
For the actual ubiquitin protein?
Yeah,
The K48 is the ubiquitin protein. So if like
or the site on the other protein that's being graded.
on the NSP5
Yeah,
protein that's
yeah.
being
Is
degraded.
that the K48?
No, that's like the viral protein. That's the like protease that's being used to cut up the coronavirus, like OC43.
So you found one site that seems to be promising. Are there multiple? Like what, are there multiple ubiquitination sites? Or
There's definitely a chance there are multiple ubiquitination sites. The one that we have does look promising, but we're also open to other possibilities.
and would you consider, mutating multiple sites to test that out?
Yeah, we've actually mutated like 5 sites at once to see if it would like have a large effect. So yeah, we're doing that. We're open to that. Anything else?
We're running a little bit ahead of schedule, so it's 9 o'clock right now, and Griffin's going to go at 9:45. I know some people are tuning in right, on time, so we've got a couple-minute break right here already. Okay, so have a little more coffee. There are restrooms by the elevators over there if you had too much coffee. Hey, Griffin, I just made it so you can share. Hi, Matt.
Awesome, thank you.
Over here somewhere.
Hey, how's it going?
Good, good. Yeah. I just don't want to get too far ahead of ourselves. Like that, the worry is you're going to get too far behind, but yeah. We need more questions.
Yeah, the first thing is a lot of questions, that kind of.
Yeah. Griffin, we're gonna give them about 2 more minutes. Where are you right now?
I am in my hotel room in Marco Island, Florida, staring out at the ocean. So I'm pretty. I was gonna give the presentation like with my back to the ocean,
Yeah.
but I couldn't get the contrast to work
Oh,
because
yeah.
I mean, hold on, let me just.
I feel like that would be pretty cruel. Oh my gosh.
Yeah.
So
So it's pretty nice,
That is
but
nice.
yeah, not traveling for the past 3 years, we built up a lot of Marriott points and we cashed them in all at once.
I got it.
Yep.
Got it.
But, yeah, so I got a nice view, but I couldn't get it otherwise.
I think you'd make everyone jealous.
Yeah.
It's currently like, I don't know, it feels like 100% humidity in Pittsburgh right now, so.
So you're like in the same boat generally, but
Not as nice of a view of pavement, though.
yeah, that's probably true. Although I am kind of sad that I can't be there like in person because like, you know, it'd be a little bit cooler to get up there and present in front of everybody. I feel like I've been virtual at like the worst times, but you know. Dr. Duncan, it looks like we're wearing very
Yes,
good.
we go ahead and have a seat. We'll get started in just a second with Griffin. Griffin, I'm gonna put the and Matt, I'm gonna put the speaker, the microphone onto my computer, and let's see how that works. Griffin, are you sharing right now?
No, I'm not. I think you're still sharing.
Oh, oops. Oh, right. Okay. Matt, are you in the lab?
Yes, actually.
Not in Florida. Okay.
No, I'll probably head over in a few minutes, yeah.
Griffin, let me know when it comes up because I'm still not seeing it.
Oh, do you want me to share now? Okay,
Yeah,
I thought Matt was going to do his introduction.
Make sure it's working.
Yep. And wait, hold on 1 second. Now it should work.
Okay, hold on now. It's only on my computer, but not on the. Shoot, how do we do this? Screen. Did I take it away from you, or can you still see yours, Griffin?
Yep.
All right, I am not seeing it here. Let's see. Somehow it's completely disappeared. Hold on, I'm gonna stop that. Oh, maybe if I just do this. That's all. Okay. All right, let's go ahead and do it, you guys.
All right, cool. So good morning, everyone. My name is Matt, and I have the pleasure of introducing my student, Griffin. You can hear me, right? Okay. This is now the second year that I've worked with Griffin in the lab of Dr. Yannis Servantinakis, and I've really enjoyed watching him grow scientifically over these past 2 years. And I've also learned this summer that Griffin's joining Pitt's undergraduate computational biology program in the fall as a freshman, which I think really suits his interests and skills that he's developed both inside and outside of the lab. And, in between all the classes and studying, I was excited to hear that maybe he'd like to continue working on this project in the fall to really take it to the next level. Griffin's been immensely busy this summer, helping out with quite a few projects, including designing and organizing a microfluidics activity for other high school students in the LEADS program. But today, we'll be talking about his main project this summer, where he's been looking at how the organization of tumor cells and fibroblasts in a tumor may determine how these tumor cells respond to targeted therapies. So all yours, Griffin.
Thanks, Matt. All right. Hi, everybody. As you heard, my name's Griffin Hurt. Can everyone hear me all right?
Yep,
Working
sound
all
good.
right? Awesome. Thank you. All right. So my project was on the spatial analysis of fibroblast-mediated drug resistance in HER2-positive breast cancer. All right. So the motivation behind this project was, so we know that fibroblasts, which are a special type of skin cell, contribute to disease progression in a variety of different ways. So you can see in this image we have here on the right side of the slide, all the different ways that cancer-associated fibroblasts affect cancer treatment. And so one of the big things is that they can influence therapy resistance. So in HER2-positive breast cancer, drugs eventually lose their effectiveness. And this is possibly caused by the fibroblast tumor cell interaction. They also affect tumor cell survival and proliferation. So proliferation is how much a cell is replicating and how it's continuing to live on as opposed to just remaining dormant. So Marusik et al., which is another paper that looked at fibroblast spatial interactions with tumor cells, demonstrated there's greater density of proliferating tumor cells closer to fibroblasts as opposed to further away from fibroblasts. And we've also seen in vivo that mice injected with HER2-positive breast cancer tumor cells, more proliferating tumor cells are close to the fibroblast-rich stroma, which surrounds the tumor, as opposed to in the center of the tumor. So we're seeing that there's a lot of interactions between tumor cells and fibroblasts spatially. So we see that that relationship exists. So we want to create a model of fibroblast to tumor cell interactions in the tumor microenvironment in order to develop more effective treatment. And one of the ways that we're developing that model is to establish a spatial relationship between the fibroblasts and the tumor cell proliferation. And the way we're going to measure tumor cell proliferation is with a marker called Ki67, which degrades when cells stop growing. So it is in higher numbers whenever cells are proliferating a lot as opposed to when they're not. And so we're going to investigate 3 different spatial metrics. We're going to look at the distance to the closest fibroblast, similar to what the other paper looked at. We're going to look at average distance to fibroblasts, so what the distance is to all the different fibroblasts and what the average distance is. So if it's close to a lot of fibroblasts versus not very many. And also similar tumor microenvironment models have observed connections between what's called Voronoi neighbor frequencies and other data. So a Voronoi diagram, I have an example of one here on the slide, is basically when you have a bunch of points on a plane, you fill in all of the pixels that are closest to one point, like that's considered a single polygon. So like, for instance, if I get my laser pointer out here, you can see that, like, for instance, this point here, all of the pixels closest to that point and no other points are colored this light green color. And then you could say that its Voronoi neighbors are all of these other polygons that are directly adjacent to this polygon. So you can see how the Voronoi neighbors might affect stuff. And so just to get that a little bit more solidified, here's some diagrams. So blue is our selected tumor cell, green is our other tumor cells, and then these orange triangles are the fibroblasts. So the closest distance is just if you're looking at this tumor cell, and this is the closest fibroblast, you just look at the singular distance, that's your closest distance. Average distance, you look at all the different fibroblasts surrounding the selected tumor cell, and you find the average of all these different distances. And Voronoi neighbor frequencies, if these are all of the neighboring cells, you have some fibroblasts, some tumor cells, you basically add up all of the different types of cells, and you divide them by the total. So if we have 2 fibroblasts here out of 6 total cells, it's Voronoi neighbor frequency for fibroblasts is 0.333 and 0.666 for tumor cells. And so that's how we're looking at Voronoi neighbor frequencies. So the goals of this project specifically are to determine whether fibroblast distances, both closest and average, influence the individual tumor cell proliferation for the EFM192 cell line. EFM192 cell line is a type of HER2 positive breast cancer cell line. We also want to investigate the effect of fibroblast-Voronoi neighbor frequency on cell proliferation, so the metric I talked about. And we finally want to develop an image analysis pipeline with a spatial component. So we want to look at the distances and how they can basically be used to establish other relationships with other cell lines or different interactions in the tumor microenvironment. So our preliminary image analysis pipeline, we took photos of the EFM192 tumor cells expressing GFP, which is green fluorescent protein. So these are breast cancer tumor cells that glow, so we can see them easier under the microscope. And we also used AR22 fibroblasts, which is just a type of fibroblast. And we gave them different doses of lapatinib, which is the cancer drug. And we imaged them after 96 hours. And we looked at Ki67 and hoaxed, which Ki67 is the proliferation marker, and hoaxed, is a nuclear marker, so we can see the cell nuclei pretty easily. And so then after we took all those images, we loaded them into the Cell Profiler software, which is a software that allows you to build an image analysis pipeline, and we segmented them by hoaxed. And then we classified each one of the identified objects as a tumor cell or a fibroblast based on the intensity of the green fluorescent protein, because only tumor cells express GFP. And then we took the object location and the intensity data and we exported it into a spreadsheet for further analysis. So here at the bottom, you can see, here's one example image that is a microscope field of the hoax channel. And then we ran it through Cell Profiler. And you can see that those that are outlined in green are tumor cells, how they're all kind of clustered together. And those outlined in red are the fibroblasts that are kind of out floating on their own. And so then we actually went through and did the spatial data analysis in a software called MATLAB. So we loaded in the data, and then we measured the tumor cell to fibroblast distances, and we also created the Voronoi diagrams. We looked at the closest fibroblast distance, the average distance to a fibroblast, and the neighbor frequencies for fibroblasts in the Voronoi diagram. We also plotted histograms for the Ki67 intensity against each metric for the non-lapatinib treated, so that was just cells and DMSO, and the lapatinib treated with all these different concentrations of drugs, tumor cells, and then we also calculated the correlation coefficients between each one of the metrics and the proliferation, which is the Ki67 intensity. So here are the histograms for the fibroblast neighbor frequency. We can see that generally they show a negative trend, and you can also see that in the drug-treated tumor cells, it generally had a lower Ki67 intensity, which indicates that obviously cells are proliferating less when they're treated with the drug. But this is actually contrary to what we were hypothesizing, because the more fibroblasts that surrounded the cell, the less likely the cell was to proliferate, which was interesting in both non-drug treated and drug treated tumor cells. And then here's an example of the distance to the closest fibroblast and the Ki67 intensity. And we see a positive trend between these two. You can see that on the left, generally the cells have a greater Ki67 intensity as opposed to on the right, which is the drug-treated tumor cells, which also makes sense. So less proliferation in the drug-treated tumor cells as opposed to the non-drug-treated tumor cells. But this positive trend is actually contrary to what we would expect as well, because we would expect cells that are closer to fibroblasts to proliferate more as opposed to proliferate less, but we see the opposite trend here. And here's our last metric, the average fibroblast distance. So the average distance to a fibroblast associated with Ki67. We can see that generally once again, the drug-treated tumor cells proliferate less and the non-drug treated tumor cells proliferate more, but the correlation between these two is not significant. So we did not graph a trend line, and no, there's insufficient evidence to support a connection between these two metrics. So here's the correlation coefficients for each metric in a bar graph. So the closest fibroblast distance for non-drug treated tumor cells, the correlation coefficient was 0.0446. And for the drug treated tumor cells, it was 0.073. These were both found to be significant against Monte Carlo simulation. The average fibroblast distance, the trends were not found to be significant. And the correlation coefficient between the fibroblast-Voronoi neighbor frequency and Ki67 intensity was found to be negative, negative 0.0641 for the non-drug treated tumor cells and negative 0.0765 for the drug treated tumor cells. So in summary, while proliferating cell density appears to increase when closer to fibroblasts, the opposite trend appears to be true for individual cells. So tumor cells with a fibroblast closer to them tend to proliferate less. That's individual tumor cells. And tumor cells with a greater number of fibroblasts that surround them tend to also proliferate less. And these trends have a greater effect on tumor cells that have been treated with lopatinib as opposed to cells that have not been treated. So there's a couple different possible ways to explain this phenomenon. One possible explanation is that tumor cells and fibroblasts are competing for the space around proliferating cells. So when a cell is proliferating a lot and all of its daughter cells are forming right next to it, you can imagine that those are pushing away the fibroblasts or limiting the amount of fibroblasts that can be directly adjacent to a cell that's proliferating a lot and producing a lot of offspring, while a cell that is not proliferating as much can get closer to those fibroblasts. So that might be one possible explanation. Or another possible explanation is that fibroblasts secreted growth factors and cytokines diffuse quickly in vitro. So in the medium, they disperse very quickly, as opposed to in vivo, they have to pass through the denser extracellular matrix. And the reason why we didn't perform this experiment in vivo initially is because it's very difficult to identify the different types of non-cancer cells. So what's a fibroblast versus the other type of non-cancer cells that are present in the tumor or around the tumor in the microenvironment. And looking towards the future, obviously we want to perform the same study in vivo because it might result in different correlations between the fibroblast distance and the tumor cell proliferation because of the reason I explained with the density of the extracellular matrix as opposed to just going straight through the medium. We also want to investigate if average distance or neighbor frequencies affect proliferating cell density. So similar to that other paper, looking at cell densities as opposed to proliferation of individual cells might indicate more about how the cells are growing as opposed to just in any individual cells because of the competition I explained earlier. We also want to standardize an image analysis pipeline that includes spatial metrics. So we want to look at Voronoi neighbor frequencies, which we've seen to be useful here as the correlation was significant. And another metric that we can use is the randomness of tumor cell fibroblast interaction locations. There was another paper that published looking at basically spatial randomness. So where certain things are distributed and how random they are, as opposed to having a correlation, almost like a spatial correlation coefficient. And finally, we want to utilize spatially aware machine learning, such as convolutional neural networks, maybe random forest classifiers, similar to what I looked at last year with, we used a software called Elastic, which basically builds random forest classifiers for identifying pixels in an image. And so we want to see if we can use that type of spatially aware machine learning in order to analyze the relationship between fibroblasts and tumor cells. So here are my acknowledgements. Principal investigator was Dr. Yannis Servantonikis. My mentor, who you heard from, was Matt Poskis. Dr. David Boone was the head of the UPMC Hillman Academy. And Dr. Duncan, who you've heard from a lot today, was the head of the Technology Drive X site. And thank you to the UPMC Hillman Cancer Center and the University of Pittsburgh as a whole. And here are my citations. Thank you.
That was great, Matt. People can ask questions and I'll repeat them back to you. Who's got some questions for Griffin? Everybody got that completely? Nothing? Griffin, what can you say about just the contact inhibition that the cancer cells might naturally have? Do they have contact inhibition or not? Could that explain anything that you're seeing when they're surrounded by the fibroblasts?
Contact inhibition meaning like when they're surrounded by more cells, the less they tend to proliferate?
Yeah, just, for some cell types, when they start to grow, if they start to have neighbors around them, they just decide to stop.
Well, so we didn't really get to investigate that in this study just because the nature of the closest thing to that would be like the Voronoi neighbor frequencies. But given the kind of way that Voronoi diagrams operate, they cancel out any empty space. So we didn't look at empty space frequencies around the cell. So perhaps that's something else we can investigate in future studies to see if the empty space around the cell also has an impact on the proliferation. And if this is just overcrowding and the cells just stop proliferating because they have so many neighbors, as opposed to it being a specific thing with fibroblasts.
Got it. Oh, there's one question. Yep.
Were you able to test with like cytokines with treatment beyond Serous? I know an earlier talk that the very
Griffin, were you able to treat with any other cytokines or investigate other mechanisms that could lead to like this motility?
We did not. This was just solely to see if there was a relationship between the tumor cells and the fibroblasts. But going forward, once again, that's other things that need to be looked at in order to establish a full model of the tumor microenvironment for HER2 positive breast cancer cells.
Got it. All right. Well, thank you.
All right. Thank you.
Griffin, enjoy vacation, everybody says.
Thank you.
Next up, we've got Priyasha, and she's going to be introduced by our mentor who is in California right now, and she's tuning in by Zoom. So she'll go ahead and do this, then we'll hook up Priyasha. Floor is yours, Isabel.
Thank you. Good morning, everyone. My name is Isabel. I am Priyasha's mentor in the Vascular Bioengineering Laboratory. Priyasha joined about 7 weeks ago, as all these students did, with a big excitement to do some research in our lab, and especially focus on cardiology, pathology, or gynecology is what she was really interested in coming in, which was a perfect fit for our lab. We focus mainly on vascular research, but recently began some endometriosis research. And I'm very excited for Priyasha to share her hard work with you and talk with you about some of the things she learned and her ability to troubleshoot throughout this summer, especially. So thank you so much for all your hard work, Priyasha. We're so happy to have had you in the lab.
You can wire yourself up. Share. In there. Screen share. Make it smaller.
All right, hello everyone. My name is Priyasha Itani, and this summer I had the wonderful opportunity to work with the Vascular Bioengineering Laboratory on a project pertaining to endometriosis angiogenesis. Now, before I begin, I wanted to take a quick poll of this room. How many of us in here are aware of what endometriosis is? All right, well, that's actually a lot of people, but but for me, I had never heard about this disease until 6 years ago when my mom was actually diagnosed with endometriosis, and I had nearly lost her, unfortunately, but she's here now, so we're all good. But that being said, endometriosis, despite being a common health problem for a majority of women, endometriosis affects approximately 190 million women worldwide. It is still one of the most understudied and underfunded diseases in the field of research today. And with that, with this lack of knowledge that we possess on endometriosis, it creates a challenge for not just patients, but physicians and scientists alike. Endometriosis, in scientific terms, is defined as the presence of endometrial-like tissue outside the internal uterine wall, and it manifests through the presence of lesions that persist throughout the peritoneal or pelvic cavity. Now, these lesions are known for causing common symptoms such as chronic pelvic pain, excessive bleeding during menstruation, and infertility. And because of this lack of knowledge that we possess on it, diagnosis proves to be difficult, often having a delay of 7 years and requiring laparoscopic surgery. Surgery is also used as a form of treatment in which diseased tissue will be excised from a patient with endometriosis. However, hormonal therapies are also currently being used as they are suggested to delay symptoms. And one of the most important things to note is that endometriosis has an extremely high rate of reoccurrence. In all of this, the problems that we experience in diagnostics, treatment, and eventual reoccurrence results from the lack of knowledge that we possess on how the disease develops. So the field's main theory of endometriosis pathogenesis, or its development, is based on the idea of retrograde menstruation, which proposes that endometrial tissue is shed into the peritoneal cavity via the fallopian tubes. However, this theory possesses one major flaw, in that 90% of premenopausal women retrograde menstruate, yet only 10% end up developing endometriosis. So there's some discrepancy there. And the big question arises in what exactly causes this discrepancy. The Vascular Bioengineering Laboratory proposes that the main variable is the idea of endometriotic conditions that leads to this 10% of women developing endometriosis. And the two main mechanisms within endometriotic conditions is one, immune dysregulation, in which it's believed that macrophages create the perfect environment for endometriosis to develop, as well as the hormone estrogen. Now, estrogen is found to be elevated within endometriotic patients, and thus, for the purposes of this research study, we will be diving into how estrogen affects endometriosis. In specific, we'll be using the synthetic version of estrogen, 17-beta-estradiol. Now, the objective of this research study was to study how increased levels of estrogen will affect the development of endometriosis. And the way in which we measured how endometriosis develops is by looking specifically at angiogenesis. Now, angiogenesis is defined as the formation of new blood vessels. And if you think about it, it would make sense. Tissue from areas where tissue is growing in areas where they're not supposed to be, and thus blood vessels are going to form around those regions to supply those cells with some nutrients. And likewise, using previous literature, we also found that endometriosis is believed to be an angiogenic factor, meaning that it induces angiogenesis through increased proliferation of endothelial cells. Now, endothelial cells are the cells that make up the internal lining of blood vessels. And so this dives us into the main hypothesis. Now, for one, we hypothesize that increased levels of estrogen will result in the increased proliferation or growth of endothelial cells, which is going to be an indirect measurement of angiogenesis as a whole. And we also hypothesize that higher vasculature will be present within endometriotic tissue, alluding to the idea of increased angiogenesis leading to the development of endometriosis. In order to test these two hypotheses, we looked at an in vitro cell culture approach, as well as an ex vivo histology approach. Now, if you look here, this is our overall experimental setup. So in the first portion, we used an in vitro approach in which we had conducted a proliferation assay in which we had looked at the different proliferation or growth rates of human coronary artery endothelial cells that were treated with our different experimental groups. Supplemented basal media being our positive control, Dilbeco's modified Eagle media being our negative control, and the different quantities of estrogen that we gathered based on previous literature. We also then seeded endothelial cells on top of a collagen gel and treated them with the same experimental groups to look at to measure the different quantities of cells based on the treatments that they received. Then, if you follow me over here to the ex vivo approach, we had taken tissue samples from patients that did not have endometriosis, so the control, as well as patients that were diagnosed with stage 3, 4, or 4 of endometriosis. And we essentially used hematoxylin and eosin staining to look at the different structures of the tissue samples. All right, so after we had conducted the proliferation assay, we originally conducted the proliferation assay using 5,000 cells per well. And we found that, if you look over here, we found that there was an unreliable relationship between the positive and the negative control, meaning that we essentially couldn't use this data. So we instead redid our protocol using 10,000 cells per well. And this time we did see that there was a reliable relationship between the positive and the negative control. And although statistically insignificant, we saw that there was a general trend of increased proliferation as we increased the amounts of estrogen that we treated the cells with. Moving on to our collagen gel. we first observed that the cells had begun to display core formation. Now, core formation is when endothelial cells begin to line up next to each other, mimicking the formation of the endothelium of blood vessels. And what this essentially told us was that collagen as a whole was a good substrate to use. It meant that the endothelial cells liked using collagen, which is going to be prevalent later on in our future work section. As for the results of the collagen gel, we found that cell quantities did increase when we treated the cells with 1.5 nanograms per milliliter of estrogen in comparison to the negative control. And we also found that cell quantity significantly decreased in cells that were treated with 4 nanograms per milliliter of estrogen in comparison to the 1.5. Again, this is indicative of previous literature and supports the idea of proliferation and endothelial cell quantities increasing as estrogen increases. Now, as per the histological analysis, we had first taken 2 different samples from the same patient that was diagnosed with stage 4 of endometriosis. And we compared it to a control samples. Now, if you look here to better orient ourselves, the circles with blue lining and if you look over here, pink, are described as blood vessels. This is how H&E stain picks up blood vessels. So the pink would be red blood cells inside, and then the outer layer is going to be the outer layer of the blood vessels. So what we found to be interesting was that endometriosis presents itself in various ways. It can either look as densely packed with cells like this, and it can also look similar to that of the control. Now, if I didn't have these labels here, you would probably think that these 2 would be the control. And this one would be the diseased. And if anything, this puts into perspective how difficult it is for clinicians as a whole to characterize and classify endometriosis by looking at their lesion structure. Likewise, the data that we had gathered from this analysis by quantifying blood vessels by looking at the mean area and the percent area of blood vessels in each tissue sample showed that endometriosis vasculature varies on all different levels. Again, one tissue sample with endometriosis can have tons of blood vessels, the other can have a minimal amount. So overall, in summary, what we found was that estrogen A does indicate increased proliferation of endothelial cells, indicating that it is in fact angiogenic. And it also showed us that although vasculature varies in endometriosis, overall, we would have to elaborate further with more research in that field. As for the future works and the future directions of this research study, we first want to study the second portion of endometriotic conditions by looking at how macrophages or immune dysregulation affects the development of endometriosis. We also want to look at how endometriosis develops in relation to endometriotic cells or immortalized endometriotic cell lines, 12Z cells. And finally, the final hope of this project is to incorporate both endometriotic conditions as well as eventually what we will test with 12Z cells into building a microfluidic device that can then mimic the environment that occurs in the uterine cavity when endometriosis develops. So the impacts of this project as a whole is to improve how endometriosis develops, as well as characterizing new ways in which we can look at lesion structures. And eventually, the hope is that with this microfluidic device, we can individualize patient studies and hopefully look for new ways to study potential treatments. And finally, I would like to thank the Vascular Bioengineering Laboratory as a whole, specifically Dr. Vorp, as well as Dr. Weinbaum, and then my wonderful mentor, Isabel Chikanovsky, as well as the Brown Lab for helping us with the histological analysis portion of this project. And then, of course, this goes without mentioning, but thank you to Dr. Duncan and Serafina and the entirety of Hillman Academy for offering me this opportunity. All right, thank you. Good questions? Pete?
You showed us a really cool figure about like the cord formation. Can you talk to the importance of cord formation in endometriosis, what that means in these states?
So endometriosis with port formation, it's indicative of a higher level of angiogenesis, meaning that because we're only testing with endothelial cells in this case, this is what's going to be, it's going to indicate angiogenesis beginning to form. And then angiogenesis as a whole, you have more blood supply to endometriosis, which can then result in the cells being able to proliferate more because they have more nutrients available. Sophia?
So I saw like the future directions you're looking at using like microfluidics. So are you looking at like the interaction between the cells or more like the formation of like the blood vessels through the gel?
Well, the microfluidic device is supposed to essentially model endometriosis angiogenesis, but we are planning to look at how angiogenesis is affected, for example, by the cell signals that can be sent by the 12Z cells, and then also the impact that estrogen and macrophages would have. All right, is that it? All right, thank you.
Whoops.